In the name of Allah, the Beneficent, the Merciful. Genes don't die, they are passed down generation after generation. So, here you can see there is a baby, and genes have been passed to him from father. These were passed to father from the grandfather, to grandfather from the great grandfather, and to great grandfather from the great great grandfather, and this sequence continues till the great ancestor. To correlate gene expression to clinical conditions, let's see the patients in the waiting room. So, there is a teenage girl who is discovered to have increased levels of fetal hemoglobin, and you are required to find the likely biochemical mechanisms responsible for this condition. In the side panel, the part of chromosome 11 with locus control region for beta globin genes is shown as a hint. Next, there is a boy with Klein-Wardenberg syndrome waiting for treatment of congenital deafness, and you are tasked to dig out the underlying biochemical cause of this syndrome. Next, there is a man who is prescribed gemfibrozil for treatment of high levels of plasma triglycerides, and you are required to explain how this xenobiotic is going to influence gene expression in this man for lowering of the serum triglycerides. Next, there is a boy with Prader-Willi syndrome, and its biochemical basis is asked. A girl with holoprosencephaly is awaiting treatment for cleft palate. At the level of gene expression, its biochemical explanation is required. Next, there is a patient with vaso-occlusive episodes, which means that any stress like high altitude hiking or any infection or dehydration can trigger episode of occlusion of capillaries leading to pain. He is prescribed hydroxyurea. Biochemical rationale for this treatment is asked, and as a hint, synthesis of globin chains before and after birth is shown in the side panel. Next is a patient with history of multiple tumors. He possesses a mutated p53 gene. Biochemical changes in this man at the level of gene expression are asked. Still further, there is a girl with iron deficiency anemia. You are tasked to dig out the biochemical alterations at the level of gene expression in her serum ferritin levels, along with the adaptations in her transferrin receptors. There is a boy with thalassemia major awaiting for blood transfusion. Next, there is a patient with retinitis pigmentosa who cannot visualize his peripheral fields of vision and has also developed night blindness, and at the level of gene expression you are required to dig out the underlying biochemical cause. So all these patients have gathered there to stimulate and channelize your thinking towards the objectives of this presentation. By the end of the presentation, you will be able to solve all these problems. Compared to prokaryotes, the eukaryotic gene expression is regulated by a wider range of processes. Since genes are expressed through proteins, or the product of gene expression is a protein, so whenever we talk about gene expression, we are talking about protein synthesis, starting from the level of gene transcription, messenger RNA formation, and its translation into proteins. Why eukaryotic gene expression regulation is different from prokaryotes? Because compared to prokaryotes, eukaryotic genome is complex because there is a nuclear membrane surrounding it. Therefore, it is regulated by a wider range of regulatory processes compared to prokaryotes, though the basic theme is same. That is the binding of trans-acting factors to cis-acting elements on the DNA. But prokaryotes lack STFs or specific transcription factors. Not only that, the operons, which consist of a single promoter controlling sets of genes, are not found in eukaryotes. Each eukaryotic gene has its own promoter. There are no operons in eukaryotes. Still further, the eukaryotic cells undergo differentiation. Eukaryotic DNA is complex with histone proteins. Mammalian genome has 1000 times more DNA than E. coli. Most mammalian cells are diploid. Major part of the mammalian genome does not code for proteins. Though some mammalian genes are unique like bacterial genes and have one or a small number of copies per genome, but still others may have many copies unlike bacterial genes. Small repetitive sequences are dispersed throughout eukaryotic genome that do not code for proteins but form bases of DNA fingerprinting. Eukaryotic genes contain introns, whereas there are no introns in the bacterial genes. Therefore, in eukaryotes, the gene expression is not only regulated at transcriptional level but at multiple other levels as well. For example, post-transcriptional regulation at messenger RNA level includes alternative messenger RNA splicing and polyadenylation, control of messenger RNA stability, and control of translational efficiency. Additional regulation at protein level occurs by mechanisms that modulate stability, processing, or targeting of the protein. Organisms with more than one chromosome need to coordinately regulate a group of genes to cause a particular response. An underlying theme occurs repeatedly. A transacting protein, which functions as a specific transcription factor or STF, binds a cis-acting consensus sequence on the genes in the group even if they are present on different chromosomes. To remember difference between cis and trans-acting elements, it can be imagined that cis-acting elements are present where DNA is, as these are the DNA sequences. In contrast, the transacting elements are the proteins that board transatlantic flights to reach the DNA. Now, since it is clear that transacting elements are proteins which act as specific transcription factors or STFs, let's dissect the STFs. Each STF has a DNA binding domain DBD and a transcription activation domain TAD that recruits coactivators such as histone acetyltransferases and general transcription factors that, along with RNA polymerase, are required for the formation of the transcription initiation complex at the promoter. Although the transcription activation domain or TAD recruits a variety of proteins, the specific effect of any one of them is dependent upon the protein composition of the complex. This is known as the combinatorial control. Examples of coordinate regulation include galactose circuit and hormone response element. To start from simple, let's discuss galactose circuit in yeast and regulation of its gene expression for metabolism of galactose. This circuit comprises of a regulatory scheme that allows the use of galactose when glucose is not available. Yeast is a unicellular organism and the genes required to metabolize galactose are on different chromosomes. So, coordinated gene expression of galactose circuit is mediated by the protein GAL4. That is the protein GAL4, where GAL stands for galactose. GAL4 is an STF or specific transcription factor. It binds to a short regulatory DNA sequence upstream of each of the genes. The sequence is called upstream activating sequence GAL or UAS-GAL. Binding of GAL4 to UAS-GAL through zinc fingers in its DNA binding domain occurs both in the absence and presence of galactose. When sugar is absent, the regulatory protein GAL80 binds GAL4 like this, and it binds at its transcription activation domain and covers or hides its transcription activation domain so that it cannot recruit proteins required for transcription of the genes for metabolism of galactose. In contrast to this, when galactose is present in the environment of the yeast, it activates another player, the GAL3 protein. GAL3 allures and binds GAL80. So, binding of the GAL80 to GAL3 uncovers the transcription activation domain. So, it is able to recruit transcription factors required for transcription of genes for galactose metabolism. So, these genes are transcribed into messenger RNA necessary for the synthesis of the enzymes which are required for galactose metabolism. In contrast, glucose prevents use of galactose by inhibiting the expression of GAL4 protein. The eukaryotic gene expression may be categorized into four major themes. The first theme may be changes in the genes, which encompass loss of genes, amplification of genes, rearrangements of genes, or modifications of the DNA bases. The second theme may involve regulation at the level of transcription, which may be through chromatin remodeling or the role of inducers to stimulate the transcription of genes. Then there may be regulation during messenger RNA processing and transport, which may be through splicing, alternative polyadenylation, and or messenger RNA degradation, or messenger RNA editing, or RNA interference. Last but not the least, gene expression may also be modulated during translation of gene product, during initiation or elongation reactions of the protein synthesis. Now let's take up these themes one by one. So first of all, I will discuss changes in the genes. Now coming to the loss of genes. Well, there can occur total or partial loss of genes, and as a result, functional proteins fail to form. This patient with Klein-Wardenburg syndrome can help us in understanding consequences of gene loss. This little guy probably suffered a loss of PAX genes in the embryonic period. Normally, stepwise and coordinated gene expression is necessary for proper tissue differentiation during embryonic life. Homeobox genes, abbreviated as HOX genes, along with closely related paired box genes, abbreviated as PAX genes, produce groups of regulatory proteins called homeodomain proteins. Now, these homeodomain proteins play a major role in control of embryonic gene expression. And how they do this is that they are responsible for activating a different set of genes at a designated period in the development. Mutations in HOX or PAX genes lead to developmental errors. This boy is likely to have a mutation in the PAX gene, which has led to dystopia canthorum, pigmentary abnormalities, congenital deafness, and limb abnormalities. All these tissues affected in this boy are derived from embryonic tissue in which PAX3 gene was supposed to form homeodomain regulatory proteins which were supposed to activate genes for development of inner corners of the eyes, pigments, hearing, and limbs. So, since these genes were not activated at the proper times, these proper proteins were not formed, and he has presented with these abnormalities. Second sub-theme of changes in gene is gene amplification. Resistance to methotrexate may be coined as an example of gene amplification. Methotrexate, shown in blue color over here, is an analog of the folic acid, shown in green. Methotrexate exerts its effect by interfering with dihydrofolate reductase enzyme because this enzyme mistakes it for folic acid. However, methotrexate therapy may lead to amplification of the dihydrofolate reductase gene. So, due to amplification, hundreds of copies of the dihydrofolate reductase may be produced. So, dihydrofolate reductase is produced in large amounts, and this overcomes the effect of binding of methotrexate to the few dihydrofolate reductase enzymes. Our next sub-theme for changes in genes is movement of DNA segments. This can be explained by immunoglobulins kappa chain formation in undifferentiated stem cells in the bone marrow. The genes for kappa light chains of immunoglobulins are divided into three segments. The V or the variable segment that encodes the first 95 amino acid residues of the variable region. The J or joining segment which encodes remaining 12 amino acid residues, and the C segment that encodes the constant region. The genome has approximately 300 V segments, 4 different J segments, and 1 C segment. As stem cells in the bone marrow differentiate to form a mature lymphocyte, 1 V segment and 1 J segment are brought together by specialized recombinations, which involve programmed DNA deletion, where the intervening DNA is discarded. Since there are 300 V segments and 4 J segments, so multiplying 300 by 4 makes it 1200. So around 1200 V to J recombinations are possible. Not only that, additional variations in sequence may occur at the V to J junction, which increases the variation by a factor of 2.5. So multiplying 2.5 by 1200 makes it 3000. So 3000 V to J combinations in primary transcript are possible. The final joining of V to J segments in combination to the C region is accomplished by RNA splicing reaction after transcription. Just beyond each V segment and just before each J segment lie recombination signal sequences. These are bound by specific proteins that catalyze double-strand breaks between signal sequences and the V or J segments to be joined. So that is the final light chain polypeptide of the kappa chain, which after folding is incorporated into the immunoglobulins as the light chain. The genes for lambda and heavy chains are also formed by similar process. Heavy chains have more gene segments. The heavy chains are shown by orange color over here. So heavy chains have more gene segments than light chains. They can make around 5,000 possible combinations. Any heavy chain can join with any light chain to generate an immunoglobulin. So multiplying 3000 by 5000 makes it 1.5 into 10 power 7 possible immunoglobulin combinations. And not only that, further diversity is generated by the mutation rates. Transposons are mobile DNA elements. They move in an essentially random manner from one side to another on same or different chromosomes. This movement is mediated by transposase, which is an enzyme mediated by transposon itself. As a result, two types of movement can take place. Direct movement, where a transposase cuts and inserts a transposon at a new site. So here you can see that transposase enzyme has produced nicks in the DNA, and now it is going to insert the transposons in the space which it has created by the nick. This is one kind of the transposon movement. The other is the replicative movement, when a transposon is copied and the copy is inserted somewhere else while the original remains in its place. Next sub-theme for changes in genes is modification of DNA bases. Methylation of CpG islands can be coined as an example here. CpG site on genome stands for cytosine and guanine, appearing consecutively on same strand of nucleic acid, where P stands for phosphodiester bond. CpG islands are stretches of DNA approximately 1000 base pairs long. They have a higher CpG density than rest of the genome and often not methylated. 70% of gene promoter regions reside in these islands, especially the promoters of housekeeping genes. The post-synthetic addition of methyl groups to the fifth position in cytosine, so that is a cytosine and that is its fifth position where methyl group may be added by methyl transferase from S-adenosyl methionine. So when these methyl groups are added, it alters the appearance of the major groove of the DNA to which the DNA binding proteins are supposed to bind. So DNA methylation regulates gene expression by two mechanisms. First, by recruiting two types of proteins by these methyl groups. These include repressive methyl binding proteins involved in gene repression and chromatin remodeling proteins that condense DNA, making it unavailable for transcription. So these methyl groups are going to recruit these proteins. Not only that, the second mechanism is inhibition of the binding of the transcription factors to the DNA. Methylation of CpG islands results in stable silencing of gene expression, because now the DNA is not available for transcription and it is not available for the binding of the transcription factors. So during gametogenesis and early embryonic development, CpG islands undergo differential methylation. This is the basis of imprinting. When a gene from only one parent is expressed. To sum up, transcriptionally active genes are less methylated, whereas DNA hypermethylation silences gene expression. Globin genes are an example. These are extensively methylated in non-erythroid cells and therefore not expressed. In contrast, they are hypomethylated in erythroid cells and hence expressed. The next major theme is regulation at the level of transcription. In eukaryotic cells, DNA is packaged in chromatin structures with the help of histone proteins, which are shown by yellow color here. Active chromatin contains histone proteins that have been covalently modified at their amino terminals called histone tails. So, genes are expressed as proteins. Proteins are formed through transcription of genes followed by translation. And for transcription to take place, tightly packed DNA needs to be reconfigured in a way that the promoter regions are exposed and become available for the transcription machinery. This links gene expression to histone proteins. The tight packing is relaxed through interference at biochemical level. The interactions between the negatively charged phosphate groups of the DNA and the positively charged histone proteins are reduced so that the desired gene region becomes accessible to RNA polymerase and the transcript. Gene activation is associated with chromatin modifications. Histones in nucleosomes undergo a variety of post-translational modifications on their tails, as you can see these colored tails dangling out of the histone proteins. Now the modifications may include acetylations, as shown by turquoise color over here, or methylations shown by the green color, or the phosphorylations shown by the red color. Further, the positions of histones may change. Histones may slide past each other, or the histones may be lost, or still further, the histones may be removed and replaced by histone variants as shown by the yellow color over here. And not only that, the histones may slide to new positions. A major mechanism for euchromatin formation is the covalent modification of the histones. For example, the acetylation of lysine residues at the amino terminus of the histone proteins by histone acetyl transferases, or HAT, which eliminate the positive charge on the lysine. Here you can see the white colored lysine hanging from the tail of the histone proteins. So when the negative charge is eliminated, the interaction between the histone protein and the negatively charged DNA ceases. Histone acetylation removes the positive charge from lysine residue on the tail of histone proteins. So here you can see that it has removed the charge, and this has led to the relaxation or decondensation of the DNA which was wrapped around the histone proteins. So that is the lysine residue, and here you can see the acetyl group that has been added, and the positive charge on the lysine residue has been eliminated. So acetylation of lysine residues on the tails of histone proteins increases transcription by three biochemical mechanisms. First, it weakens the interaction between histone tail and DNA by removing the electrostatic interaction, leading to destabilization of the nucleosome. So here you can see that there is electrostatic interaction between the DNA and the tails of the histone proteins, and addition of the acetyl groups leads to loss of this electrostatic interaction. And second mechanism is that the acetylation weakens the interaction between nucleosome, and this leads to their disassembly from higher order chromatin structure. As shown here, you can see that the nucleosomes disassemble, they become slightly disorganized, and DNA becomes accessible. Third mechanism is that histone acetylation provides binding sites for the addition of transcription factors. So here you can see that now the DNA is relaxed, and nucleosomes are not properly organized, and here the transcription factors can bind to the DNA. Removal of acetyl groups by histone deacetylases restores the positive charge and the interactions between positively charged histone protein tails and the negatively charged nucleic acid DNA. So here you can see there is a lysine residue hanging from the histone protein tail, and at the end of this residue there is the acetyl group. Now histone deacetylase removes this group and restores its positive charge and restores the tight packaging, leading to the formation of heterochromatin. The transcription associated structural changes in chromatin are collectively called chromatin remodeling. The acetyl, methyl, and phosphoryl groups not only serve as docking sites for transcription factors, so these are the transcription factors and these can dock on these groups. Acetylated histones also interact with the bromodomain proteins, whereas methylated histones interact with chromodomain proteins. Bromodomain proteins, with the help of ATP, displace histones to free up an area for transcriptions. So, bromodomain proteins with the help of ATP may cause displacement of the histones to free up the area for transcription. It is important to appreciate here that histone methylation is different from acetylation. Lysine methylation differs from acetylation in several ways. It does not affect the charge on lysine residue, whereas acetylation removes the charge on lysine residues. Secondly, lysine residue can carry 1, 2, or 3 methyl groups. Third, methylation is longer lasting modification than acetylation. Methylation of some histone lysine residues is associated with gene activation, whereas methylation of other lysine residues may be associated with gene repression. Another type of chromatin modification that affects gene expression is methylation of nucleotide base cytosine in regions of DNA near the gene promoters. Methylated CG sites or CPG islands in DNA appear to affect the recruitment of chromatin remodeling proteins that condense to silence the gene transcription. So, here it is illustrated that you can see the dangling tails of the histone proteins with the lysine residues, and these can be methylated and these can affect the gene expression. And not only that, CPG islands on the DNA may be methylated at the cytosine residues. The next sub-theme for regulation of gene expression at the level of transcription is through the action of specific transcription factors or STFs. Hormone response elements or HRE are the DNA sequences that bind the trans-acting transcription factor proteins and regulate gene expression. Further, the regulation at the level of transcription is also modulated by the binding of coactivators to the DNA binding proteins. And not only that, certain hormones affect gene transcription by acting on the cell surface receptors. To illustrate regulation of gene expression through transcription factors, the example of steroid hormone cortisol may be coined. So it is a glucocorticoid. It binds intracellular receptors. So here you can see these glucocorticoid receptors shown by green color. Binding causes a conformational change in the receptor. So here you can see that the steroid hormone has bound to the glucocorticoid receptors, and now there is a conformational change in the receptor and the DNA binding domains have become conspicuous. So now not only that, the receptor hormone complex has dimerized. The two receptors for glucocorticoids have dimerized, and now these are going to enter the nucleus. So, and they recognize and their DNA binding domains bind to the GRE. GRE is the glucocorticoid response elements, and these are just the sequences of DNA, and it is a type of HRE or hormone response element. So now, since it is binding allows the recruitment of co-activators to the transcription activation domain or TAD, and so here you can see that now the co-activators are recruited and the transcription bubble is formed. So the net result is the expression of the cortisol responsive genes. So here you can see that now messenger RNA is formed and there is protein synthesis according to the cortisol responsive genes. So the GRE can act as a true enhancer as in this case, or alternatively if it is associated with repressors, hormone response complex is inhibited and transcription does not take place. Now coming to the cell surface receptors, the cell surface receptors include receptors for insulin, epinephrine or glucagon. So here the receptor for glucagon is shown, and you can see that when the glucagon binds, it is a G protein coupled receptor and there is a conformational change in the G protein and its alpha subunit in the presence of GTP binds to the adenylyl cyclase, which is shown in the blue color here, and adenylyl cyclase then leads to the activation of protein kinase through the production of cyclic AMP, and the protein kinase A is going to phosphorylate proteins including the this pink one, cyclic AMP response element binding protein. So when it is phosphorylated, it is going to enter the nucleus and bind to the cyclic AMP response element, leading to the gene transcription of the proteins required by glucagon, in this case glucose 6-phosphatase for glycogen breakdown and PEP-CK for gluconeogenesis. Some xenobiotics like gemfibrozil bind specific transcription factors and modulate gene expression. Peroxisome proliferator activated receptors or PPARs are transcription factors that bind to DNA response element PPREs and control multiple aspects of lipid metabolism. So here you can see that blue colored gemfibrozil, it enters the cytoplasm, binds to the PPAR, and then the PPAR moves to the PPRE, which is the response element. Then there is recruitment of the proteins for the formation of transcription bubble, and this is going to affect the enzymes involved in the lipid metabolism. For example, increased expression of lipoprotein lipase may help the patient to whom which is prescribed in lowering the levels of the serum triglycerides. The next major theme for the regulation of gene expression in eukaryotes is messenger RNA processing. Eukaryotic messenger RNA undergoes several processing events before it is exported from the nucleus to the cytoplasm for use in the protein synthesis. Capping at 5' end and polyadenylation at 3' end and splicing are essential for the production of a functional eukaryotic messenger RNA from free messenger RNA. Variations in splicing and polyadenylation can affect gene expression. In addition, messenger RNA stability also affects the gene expression. Splicing reactions remove the introns and connect the exons. At the 5' end of an intron, there is a sequence G2. So this is the intron. There is a splice donor site towards its 5' end, and the intron is shown in yellow color. And this is the sequence G2. And at its 3' end, there is AG. After formation of primary transcript that contains introns and exons, 5 small ribonucleoprotein particles or SNPs mediate splicing by forming base pairs with consensus sequences at each end of the intron. This brings the sequences of the neighboring exons into correct alignment for splicing. The 5 SNPs bind to the primary transcript. After 5 SNPs bind the U1 and U4 leaf. This leads to the activation of the spliceosome in the presence of ATP. So now there is a spliceosome formation. Now the 2' OH of the branch site A. So this is the branch site A and that is the 2' OH. It attacks the 5' phosphate. This is the 5' phosphate at the splice donor site of the intron. And this forms a 2' to 5' phosphodiester bond and a lariat-like structure. Now what happens is that the 3' OH, so this is the 2' to 5' phosphodiester bond. Now this 3' OH of exon 1 attacks the 5' phosphate at the splice acceptor site, forming a phosphodiester bond that joins exons 1 and 2. So again a phosphodiester bond formation between the exons 1 and 2 which form the mature messenger RNA, and the intron is released as a lariat-like. Alternative splice sites can be used to produce different messenger RNAs. By this mechanism, calcitonin gene produces two different types of proteins. It produces calcitonin in the thyroid gland, whereas it produces calcitonin gene related protein in the brain. So how this works can be appreciated from this example. Here you can see 3 exons and the introns, intervening introns are shown by these triangles, and if these introns are removed, these 3 exons are going to join and produce a protein which will contain the information contained in these 3 exons. So that is one scenario. On the other hand, if different splice sites are used and the exon 2 is also removed along with the introns, a shorter protein is going to be produced which will contain the information encoded by the exon 1 and exon 3. So this is how alternative splicing can affect the gene expression. Most eukaryotic messenger RNAs have a tail which is shown by turquoise color over here. It consists of 40 to 250 adenosine monophosphates attached towards its 3' end. This tail comprises of adenylates or adenosine monophosphates. So the pre-messenger RNA transcripts may possess more than one site for cleavage and polyadenylation. This means that these may contain more than one site for or one signal for the uh, production or the insertion of the poly A tail. So here these are the signals: double A, U, triple A. Again, you can see double A, U, triple A. These signals is these are the code that the tail may be inserted here. And when the tail is to be inserted, this messenger RNA will be cleaved at this point, and the enzyme which inserts the tail will start adding the adenosine monophosphates. So let's see that there are two signals. There may be many more signals for the tail. You can see in this transcript two signals you can see. So these are the signals for polyadenylation. Polyadenylation means insertion of the poly A tail, and these are the potential cleavage sites. Now any one of these sites may be used for the insertion of tail. So if the first site is used, a comparatively smaller messenger RNA will be produced and the tail will be added. So cleavage will occur at this site and tail will be added. Or alternatively, if the second cleavage site was used, then a comparatively longer messenger RNA may have been produced. So the result is the production of different messenger RNAs with different 3' ends, with different or altered untranslated regions or UTRs, or the translated sequence. So this is how it is going to affect the gene expression. The messenger RNA which got its tail at this first cleavage site will produce a shorter protein. The second choice, which is the second cleavage site, may produce a longer protein. Alternative polyadenylation is involved in the production of membrane bound and secreted forms of immunoglobulin M. The gene for this antibody contains two polyadenylation sites, one after the last two exons, and that is the antibody. And if this is used, the antibody will contain a hydrophobic region that anchors it to the cell membrane. So here you can see that the IgM may be membrane bound as shown here, or it can be secreted out of the cell. And how this occurs is that when polyadenylation occurs at the first site, this one, the antibody lacks hydrophobic tail and it is going to be secreted. On the contrary, if the polyadenylation occurs at the second site, the antibody which is produced will contain certain hydrophobic amino acids, and it will be these hydrophobic amino acids will anchor it to the membranes. So here you can see that mature messenger RNA will be formed, and if it is formed at the first site, then there is no hydrophobic sequence and the antibody is secreted. Alternatively, if the cleavage occurs at the second site after the last two exons, some hydrophobic amino acids will be present, and the antibodies which will be produced, these are going to be bound antibodies. These are going to be bound to the cell membrane. The next subtheme for gene regulation through messenger RNA processing and use is the messenger RNA editing. The difference between mutation and editing is that mutation is spontaneous and non-enzymatic, whereas editing is not spontaneous, it's planned and enzymatic. So when messenger RNA has been fully processed, it may undergo an additional post-transcriptional modification in which a base in the messenger RNA is altered, and this is known as messenger RNA editing. An important example of editing in humans is messenger RNA editing of the transcript for apolipoprotein B or ApoB. So ApoB is an essential component of the chylomicrons and VLDL. ApoB messenger RNA is made in the liver and small intestine. So here you can see that this is the ApoB messenger RNA. This one is the start codon, and then QAA here is the stop codon. So in the intestine, the cytosine base in the CAA codon for glutamine is enzymatically deaminated to uracil. So this codon for glutamine, what happens is that cytosine is deaminated, and once it is deaminated, it forms uracil. So this deamination creates an additional stop codon over here, and this will lead to formation of ApoB48 in the intestine, which contains 48% of the ApoB protein. The next subtheme for regulation of gene expression by messenger RNA processing and use is the stability of the RNA. A crucial factor governing genes expression is the cellular concentration of its associated messenger RNA. Messenger RNAs can be degraded by nucleases after their synthesis in the nucleus and before their translation in the cytoplasm. Messenger RNAs have different half-lives. Interferon stimulates synthesis of a 2' to 5' oligo A that activates a nuclease that degrades messenger RNA. Messenger RNA decay is stimulated by deadenylation, that is, loss of its tail, or decapping, meaning removal of the 7-methylguanosine cap. The next subtheme for regulation of gene expression through RNA processing and use is the messenger RNA interference, which causes the gene silencing either through decreased expression of messenger RNA or through the repression of translation. So messenger RNA interference is mediated by short non-coding RNA, which is around 22 nucleotides long, microRNA or miRNA. These miRNAs arise from genomically encoded nuclear transcripts, primary messenger RNA or miRNA. Now this miRNA is processed in the nucleus to pre-miRNA by an endonuclease, which is named as DROSHA. So after its processing to pre-miRNA, it is transported to the cytoplasm. There an endonuclease dicer completes the processing, leading to formation of a short double-stranded miRNA. Now a single strand, the guide or the antisense strand of the miRNA, moves and associates with the RNA-induced silencing complex or RISC. The guide strand hybridizes with complementary sequence in the 3' UTR of full length of the target messenger RNA, bringing the RISC to the miRNA. And this can result in repression of translation of the messenger RNA or its degradation by an endonuclease. The endonuclease may be argonuate, ago, or slicer. So that is the animation of the process. RNA polymerase transcribes DNA into primary messenger RNA. which is further processed by DROSHA to pre-messenger RNA, which is then exported into the cytoplasm, and here the enzyme dicer further processes it and generates a double-stranded RNA, and from this guide strand, a guide strand moves to the RISC or ribosome RNA-induced silencing complex RISC. Now it can interfere with gene expression through two mechanisms. Either it can degrade the target messenger RNA. So here you can see that the guide strand goes to RISC, and from there it can lead to the degradation of the target RNA or the repression of the translation. In age-related macular degeneration, there is excess proliferation of blood vessels in retina, leading to macular degeneration. RNA interference may be used to destroy messenger RNA for vascular endothelial growth factor A or VEGF-A, which is responsible for vascular proliferation. So that is a therapeutic application of the RNA interference. The next major theme is the regulation of gene expression at the level of translation. This may be explained by following examples. Transferrin is a plasma protein that transports iron. Transferrin binds to cell surface receptors, transferrin receptors or TFR, to get internalized and provide the erythrocytes with iron. So here you can see this is the transferrin receptor and that is the transferrin in red color. The messenger RNA for transferrin receptor has several cis-acting iron response elements in its 3' untranslated regions or UTRs. So these are the cis-acting iron responsive elements. Iron responsive elements have a short stem loop structure as seen here, and it can be bound by trans-acting iron regulatory proteins. So these are the iron regulatory proteins shown in green. When iron concentration in the cell is low, the iron response proteins bind to the iron response elements and stabilize the messenger RNA for transferrin receptor, allowing the synthesis of transferrin receptors. So when the iron is low, the iron responsive proteins are going to bind these elements, and this is going to lead to the synthesis of transferrin receptor. When the intracellular iron levels are high, the iron response proteins dissociate from the iron response elements. The absence of iron response proteins bound to the messenger RNA speeds up its destruction, resulting in decreased transferrin receptor synthesis. Next, ferritin is an intracellular protein for iron storage. Its levels are also regulated by modulation of gene expression at the level of translation. So that's the when the iron concentration in the cell is low, the iron response protein, which is shown by purple color over here, it binds to the iron response element, which is at the towards the 5' end, this loop structure, and when the iron response protein binds to it, this is the iron response protein, and when it binds to the iron response element, it prevents its use, and this RNA cannot be translated into the protein ferritin. On the contrary, when the iron accumulates in the cell, the iron response proteins dissociate, allowing the synthesis of ferritin for the storage of iron. Next is the ALA synthetase 2, which is the gamma aminolevulinic acid synthetase 2. It is a regulated enzyme of the heme synthesis. Its gene transcript also contains a 5' iron response element. Under conditions of iron deficiency, the translation of ALA2 is inhibited by binding of the iron response protein to its iron response element, which is towards the 5' side. So, in presence of low iron, IRP binds the iron. On the contrary, when the intracellular iron increases, the iron response proteins are degraded, allowing the translation of the ALA2 messenger RNA. So, when the iron is high, these are degraded and the translation ensues. The next subtheme for regulation of gene expression at the level of translation involves inhibition of translation through phosphorylation and subsequent inactivation of the eukaryotic initiation factors for translation. Here you can see the eukaryotic initiation factor 2 and its phosphorylation leads to its inactivation. Finally, protein modification may also play part in gene expression regulation. For example, proinsulin is cleaved to insulin. First of all, the pre-proinsulin in the endoplasmic reticulum is modified and the signal sequence is removed. Then, and it produces proinsulin. In the Golgi apparatus, C-peptide is removed from the proinsulin to form the functional insulin molecule. To sum up, changes in genes may be due to loss, amplification, rearrangements, or base modifications. Further, at transcriptional level, gene expression may be regulated by acetylation, methylation, hormones, messenger RNA processing, and the final checkpoint for gene expression regulation is at the level of translation. The basic theme for regulation of eukaryotic expression is the binding of the STFs to the enhancer sequence. So now coming to that, which of the following is not found in eukaryotes? Well, opron is not found in the eukaryotes. In combinatorial control of gene expression, the specific effect of recruited proteins is dependent upon the transcription activation complex, which is the choice beta. Okay, for the membrane bound and secreted form of immunoglobulins, alternative polyadenylation is responsible. So choice alpha. Epigenetic gene regulation example is the chromatin remodeling. Heritable changes in DNA without altering gene sequence may be caused by the beta hypermethylated CG rich islands. Generation of 10 power 9 to 10 power 11 different immunoglobulins from a single gene are possible because of the choice delta somatic recombination. So here is a sibling homozygous for HPS and no symptoms of sickle cell disease. So he is likely to have synthesis of which of the following globin chains. He is going to have increased synthesis of the gamma chains, choice Charlie. This sibling, in addition to sickle cell disease, has hereditary persistence of fetal hemoglobin. Individuals with HP FH or hereditary persistence of fetal hemoglobin express gamma globin chains that throughout their life are present at high levels, and these prevent against the effects of the homozygous HBS expression. Now here is the research project on mice. Cholesterol is affixed to double-stranded RNA strand. It enters the plasma membrane, binds the apolipoprotein B gene on messenger RNA, and what effect is it is going to exert on the apolipoprotein B. So it is a typical case of the RNA interference where the guide strand is going to go to the messenger RNA for the apolipo B gene. Now there's a woman with BMI of 16.5. She's presenting with easy fatigability, or how she is going to maintain her blood glucose levels. So the answer is choice C, or Charlie, cyclic AMP response element binding protein, because it will be activated by the glucagon, and this will help in the gluconeogenesis. Now there is a patient with methotrexate therapy, and he has developed methotrexate resistance due to the amplification choice alpha of the DHFR gene. Now let's revisit the waiting room. So first of all, there was a teenage girl who is discovered to have increased levels of fetal hemoglobin on routine checkup, and as hints, the locus control regions of beta globin genes were shown. So the answer is that this girl is expressing hereditary persistence of fetal hemoglobin, meaning that beta globin genes are not expressed which form the adult hemoglobin, HbA or A2. And to compensate for it, other gene families which include gamma chains are expressed. Now, what is the possible biochemical mechanism that has interfered with the expression of beta globin genes? Well, there may be any of these three mechanisms. She may be having a deletion or loss of DNA controlling beta globin gene, or beta globin gene DNA in itself may be mutated. Further, gene may have been silenced through methylation or deacetylation. Next, there is a man who is prescribed gemfibrozil for treatment of high levels of plasma triglycerides, and as we have seen that gemfibrozil binds to it is a xenobiotic. It binds to intracellular receptors and affects the gene expression for lipoprotein lipase. So that is how it is going to help this man. Okay, next is a boy with Prader-Willi syndrome. Okay, the biochemical cause may be that there are few genes which have monoallelic expression due to genetic imprinting. The gene involved in Prader-Willi syndrome is on chromosome 15 and is imprinted so that it is normally expressed only from parental and not maternal chromosome. If one inherits a paternal chromosome in which this region has been deleted, Prader-Willi syndrome results. It can also result from maternal disomy of chromosome 15. So this boy has Prader-Willi syndrome because either he has inherited a chromosome 15 from his father in which this gene was deleted, or maybe he has inherited two chromosomes 15 from his mother. A girl with holoprosencephaly is awaiting treatment for cleft palate, and at the level of gene expression, its biochemical explanation is required. So, holoprosencephaly is a common developmental anomaly of the human forebrain and midface where the cerebral hemispheres fail to separate into left and right halves. Haploinsufficiency for sonic hedgehog gene is cause of holoprosencephaly. Haploinsufficiency occurs when one copy of a gene is inactivated or deleted, and the remaining functional copy of the gene is not adequate to produce the required gene product to preserve its normal function. It is postulated that a mutation in the homeobox gene affects the expression of sonic hedgehog in the precaudal plate during the embryonic life, leading to failure of separation of the cerebral hemisphere and other abnormalities. So, mutation is the Hox in the Hox gene is going to result in the formation of aberrant homeodomain proteins. These proteins are going to affect the expression of the sonic hedgehog gene. So, one of the gene is going to lose its function, and as a result, when one functional gene is only present, it is not able to form the required proteins, and this affects the development at the level of the precaudal. Next, there is a patient with vaso-occlusive episodes, and he is prescribed hydroxyurea, and biochemical rationale for this treatment is asked. And as a hint, senses of globin chains before and after birth is shown in the side panel. So the answer is that the hydroxyurea treatment is designed to activate transcription of gamma globin gene, which is normally only expressed during the fetal life as shown here. When expressed, gamma globin genes will form functional hemoglobin tetramers with alpha globin chains, thereby reducing the effects of the mutated beta globin chains. Next is a patient with history of multiple tumors. He possesses a mutated p53 gene. Biochemical changes in this man at the level of gene expression are asked. This man has Life-Frohmani syndrome. He is likely to have inherited a mutated copy of p53. p53 is a transcription factor. It monitors the health of DNA. As you can see that on the slide, the cell cycle is shown, and S1 is the phase where there is synthesis of the DNA. So that's where p53 is going to exert its effects. If DNA damage is found, transcription factor p53 initiates gene transcription to arrest cell cycle until the DNA is repaired. And if DNA cannot be repaired, here you can see there is a black colored DNA damage, and p53 is come into action, and it is going to arrest the cell cycle until the DNA is repaired. And if DNA cannot be repaired, then p53 will lead to transcription of genes for the apoptosis and cell death, so that the cell with damaged DNA does not replicate. Now this man is having the mutation in p53, so damaged DNA is replicating, causing cancers. The initial inactivating event is impaired gene transcription by p53, which is the trigger for all the events that follow. Still further, there is a girl with iron deficiency anemia, and you are tasked to dig out the biochemical alterations at the level of gene expression in her serum ferritin levels, along with the adaptations in her transferrin receptors. So ferritin is the iron storage protein within the cells, and if the intracellular iron levels are low, as in this girl, there is no need to upregulate the synthesis of ferritin, so its synthesis will be downregulated in this girl. And iron travels in circulation bound to transferrin, so increasing transferrin receptors on the cell surface will enable this girl to transport more efficiently iron into her cells, so the transferrin receptors will be upregulated. There is a boy with thalassemia major awaiting blood transfusion. This boy is having decreased production of hemoglobin and consequently anemia. He is likely to have a point mutation in Tata box or splice junction in beta globin gene. The mutation at splice site may alter the GT sequence at the beginning of the intron or the AG sequence at its end. These sequences are must for normal splicing, and such mutations can lead to loss of beta globin gene production. So, if mutation is in consensus region of splice donor or splice acceptor site, there may be reduced ability to correctly splice, and it will result in decreased but detectable amounts of beta globin. Next, there is a patient with retinitis pigmentosa who cannot visualize his peripheral fields of vision. So, why is he's having this problem is that mutations that disrupt assembly or function of nerves are responsible for retinitis pigmentosa. Since he has the autosomal dominant type, the mutation has affected Ju4 nerve. Absence of Ju4 nerve leads to disruption in splicing activity in retinal cells, which need to synthesize large amounts of rhodopsin protein. Because of continuous cell renewal in outer segments of the rod photoreceptor, because mutation is autosomal dominant, it is possible for him to have one normal copy of gene, which leads to reduction in splicing activity.